This presentation examines Indole-3-Carbinol and its primary in-body derivative, Diindolylmethane. Presented by Brian Geniuson, research scientist, this analysis evaluates the compound through a systems biology lens, integrating molecular chemistry, biodistribution, empirical evidence stratification, and translational context. In this discussion, the reported and scientifically investigated health benefits associated with this compound will be evaluated, clearly distinguishing between human clinical data, biomarker findings, and mechanistic research. The analysis begins with the strongest areas of human evidence, including measurable plasma exposure to diindolylmethane and biomarker modulation related to estrogen metabolism, before examining additional mechanistic, emerging research domains currently under investigation. Indole-3-Carbinol, or I3C, is an indole phytochemical with the molecular formula C9H9NO and a molecular weight of 147.17 grams per mole. In purified form, it appears as an off-white solid. From a natural occurrence perspective, I3C is not present in plants as a stable compound. It is derived from glucobrassicin, a glucosinolate found in cruciferous vegetables such as broccoli, Brussels sprouts, cabbage, and kale. When plant tissue is disrupted through chopping or chewing, glucobrassicin is hydrolyzed, leading to the formation of indole-3-Carbinol. However, its translational identity is defined not merely by its dietary origin, but by its chemical behavior in the gastric environment. Under acidic conditions in the stomach, I3C undergoes rapid condensation reactions, producing several oligomeric derivatives, most prominently diindolylmethane, or DIM. Clinical pharmacokinetic studies demonstrate that intact I3C is often not detectable in plasma, whereas DIM is measurable following oral administration. This conversion is critical. Many biological effects attributed to I3C supplementation likely reflect DIM-mediated signaling rather than intact I3C activity. From a biodistribution perspective, the strongest physiological engagement occurs along the gastrointestinal and hepatic axis, where transformation and first-pass metabolism take place. Moderate distribution relevance is observed in endocrine-responsive tissues, particularly in the context of estrogen metabolism pathways. Immune and inflammatory signaling domains show mechanistic engagement, though human outcome data remain more limited. At the molecular level, indole-derived compounds are frequently discussed in relation to aryl hydrocarbon receptor signaling, inflammatory pathway modulation including NF-kappa B, and cell cycle regulatory mechanisms. It is important to distinguish between mechanistic plausibility and demonstrated clinical outcomes. Within the claim landscape framework, estrogen metabolism modulation is supported by moderate human biomarker evidence. Chemopreventive discourse and inflammatory pathway interactions are primarily supported by mechanistic and preclinical data. Direct therapeutic claims are not established. Now consider the empirical evidence layer. Human evidence supports measurable plasma detection of DIM following oral dosing of I3C, confirming systemic exposure. Biomarker studies demonstrate modulation of estrogen metabolism pathways. Animal models provide tissue distribution data and mechanistic signaling insights. Cellular studies show pathway-level modulation, including receptor-mediated signaling effects. Computational and systems modeling literature remains emerging. Empirical evidence, key points. First, human exposure to DIM following I3C ingestion is measurable. Second, the strongest validated evidence currently lies in biomarker modulation rather than long-term clinical endpoints. Third, much of the broader health discourse surrounding indole compounds is driven by mechanistic and preclinical findings. Within the validated clinical and biomarker outcomes panel, the most defensible conclusions relate to measurable modulation of estrogen metabolism biomarkers. Long-term disease outcome data remain limited, and continued investigation is warranted. Overall, current evidence supports measurable systemic exposure and biomarker-level modulation within endocrine pathways. However, long-term clinical outcome evidence remains limited, and much of the broader health narrative is supported by mechanistic and preclinical domains. The translational maturity level for this compound can be characterized as intermediate, supported by human exposure data, moderate biomarker findings, and strong mechanistic depth. Full scientific literature references are available in the video description. This content is provided for educational and scientific discussion purposes only and does not constitute medical advice. For continued evidence-based analysis of natural bioactive compounds across molecular biology, systems medicine, and translational research domains, consider subscribing.